Today we are having a great opportunity to listen to the words of great knowledgeable speaker, Dr. Tariq A. Rajasan. On behalf of IPS Academy, I welcome our honorable speaker, Dr. Tariq A. Rajasan. I request our technical team for the hearty welcome of our eminent speaker. Thank you. I request Dr. Premlata Gupta ma'am, Principal ma'am, to say a few words. Ma'am, please.
Yeah. Indeed, it is a great pleasure to have Rajasan here and honorable distinguished participants. Again, I extend a very warm welcome and good morning to all of you. I'm very happy and delighted that this is the fourth day of our faculty development program. I congratulate our organizing team for their commendable job. And I also congratulate and appreciate all the participants for their punctuality and sincerity. Now I am going to give a brief introduction of IPS Academy. IPS Academy is one of the Central India's most premier education hub. Success story of IPS Academy started in year 1994. Thereafter, IPSA grew in leaps and bounds and now grown in 3 huge campuses. The main campus is at Indore. It is spread in 60-acre land. Here we are running 71 courses under 16 colleges under one umbrella. Here, about more than 10,000 students are updating their skill, knowledge, values, and the students are mentored by more than 450 faculty members. Having value-based education is the prime focus. We are committed to promote endurance, excellence, honesty as its core value. Institute of Science and Research is one of the institutes in 16 colleges of IPSA. ISR is established in 2004. We are running 6 UG courses and 6 PG courses. We have PG in microbiology, biotechnology, mathematics, physics, chemistry, pharmaceutical chemistry. We are also running PhD programs in physics, chemistry, mathematics. Now, I am again going to hand over this to Amita.
Thank you,
Again,
ma'am.
a warm welcome. Thank you very much.
Thank you very much, ma'am. Now I give a brief introduction of Tariq A. Rajasan. He is working as professor and chief scientist in statistics at Faculty of Agriculture and adjunct faculty at Division of Agriculture Statistics, Sher-e-Kashmir University of Agricultural Sciences and Technology, Kashmir. At present, he is head of Agriculture Statistics and Economics Department. He has passed MSc in Statistics from University of Kashmir, Srinagar, and has done post-graduation diploma in computer applications. He has received his PhD from Dr. Bhimrao Ambedkar University and completed 4 software training courses. He has published 127 research papers in various peer-reviewed national and international journals and has 4 manuals and 2 books to his credit. He has 27 years of teaching and research experience. Besides being in the advisory committee of more than 200 PG or PhD scholars, 3 students has been awarded PhD under his guidance. He has organized 4 training programs at national level and has received 3 awards at national level and is executive member of various national and international societies. He has presented 25 research papers in national and international conferences. Now may I request our eminent speaker, Dr. Tariq Rajasan, to start his presentation on statistical software in research. Sir, please. Yes, sir.
Am I audible?
Yes, sir.
Is the screen visible?
Yes, sir.
I'm sharing my screen. Is it visible?
Yes, sir.
Let me start then.
Yes, sir. Please, sure.
I am Tariq Raja, and I'm going to discuss something about the statistical softwares in research. So I have some 3-4 presentations with me. It's going to be a combination of 3-4 presentations. I would like to say something about softwares first. Then I would like to show some softwares like R software, Spss software, and Minitab software. And I would like to have practical demonstrations on my screen because all the softwares are available with me. And I will try to acquaint the participants with the basics of these softwares. So let me give some idea, keeping in mind that there are some participants who are totally new to these softwares and they do not have much idea about the softwares, or I must say they are not very much well versed with softwares. Let me start from the very basics. What's the connection between softwares and statistics, and how many softwares are there, and what softwares are doing. So let me start it like this. Computers have been using for statistical processing of data since long. Processing and analysis of data involves handling massive data for tabulation and carrying out complicated computations, which consumes considerable amount of time, thereby delaying the availability of results. That means when we are having a data, there can be a data that's called big data, huge data, metadata. Metadata means data about data. If we are having some data about a human being, then further data about his head circumference, about his chest circumference, about his blood pressure value, about his TLC, DLC, and so much data about a particular person. And we are having a good amount of those individuals, then data about data means metadata. So handling big data, metadata, and huge amount of data. If we are doing it on calculator and manually, it will definitely take months. It will take, depends upon the problem, sometimes 3 months, 4 months, or more than that. So with the availability of softwares, this computational labor is over, and we can jump to the results. We can find out the results within a very limited period of time, sometimes within minutes and sometimes within seconds. I must say now with the availability of statistical packages, the computational labor has been almost eliminated. Moreover, there are, if somebody is from the discipline of statistics, there are multivariate statistical techniques, which are often complex. And so computation intends to that researchers very often shy away from using these powerful and logically correct techniques on the play that data analysis would consume a lot of time. Sometimes for the validity of results and for having valid results, we need to go for multivariate analysis. Multivariate analysis means we cannot take a single variable and deal with it. We have to take number of variables and analyze them jointly, collectively. And that makes, again, the calculation and the computation very difficult and very complex. Again, the role of software comes. Software packages have changed the scenario, and now researchers are willingly using these statistically correct and logical techniques like multivariate analysis techniques, like cluster analysis, discriminant analysis, factorial analysis, logistic regression, probit analysis, and so many advanced statistical techniques and tools. Once a researcher sends a paper in a very powerful journal of having good impact factor of NAS rating, the papers are being rejected on the play that you go for the multivariate analysis because univariate analysis is not sufficient. But on the other hand, multivariate analysis is not so easy. So that can be done only through softwares. It's worth mentioning that the statisticians have been one of the first few users of computers. In fact, the first commercial computer was used for the tabulation of US Census data. Computers are now becoming indispensable part of our life, and softwares are the backbone. Or we can say software is like a soul to the computer. Computers have crept in every discipline of human activity with desirable softwares. With desirable softwares, I mean that if somebody is working in a particular field, he is having a software available in that field. Say if a person is working as ENT specialist, he is having a software which deals with the almost all those problems which are concerned or connected with ENT. ENT means ear, nose, and throat. So a doctor, first of all, then a specialist, then a super specialist is having a software in his field. Likewise, there are softwares in, I must say, in each and every field. There are softwares for super specialists. There are softwares for musicians. There are softwares for, I must say, in every human activity. Biometrics, a core area under the grab of statistical sciences, I must say, biostatistics, a science or a study of genetic statistics involves study of humans and animals. This is a very complex field if we are working in this. So here I will try to skip so many things because I have to show a number of things. Is it okay? Am I audible? Is it going on nicely?
Yes, sir.
Okay. I must say, with the advent of user-friendly software packages, the computational work has eliminated and is now possible to use logically correct statistical techniques for processing of data. The data entry, storage, retrieval of data in desired format, making cross-classified tables and applying statistical analysis and procedures has become all too easy with the availability of software packages. Now let us start where from the software packages came, what's the basics of these softwares. Computer software for statistical needs has advanced considerably, starting with BASIC, Fortran, Algol, COBOL, Pascal, and so many other basic programs. BASIC is beginner's all-purpose symbolic instruction code. Fortran is formula translation. This is a talk of some 30 years, or I must say 35 years before, people were using basics for programming. People were using Fortran. People were using Algol. Then came another like C, then came C++, usual C++. So this packages, software, or I must say the programs have changed from very basics to now there are some good statistical softwares like, I must say, we are having softwares for statistical needs, MStat. We are having GenStat. These are the statistical softwares. We are having SysStat. We are having IndoStat. Minitab, Spss, or software or language, Statistica, GLIM, Sudan, PCCAR, SPSS, etc. And there is a software that's called SAS, Statistical Analysis System. So it's not possible to describe all the features of all these software packages. We will, however, try to give you some basic idea and some basic concepts about few of the softwares which are mostly in use. Some of the software packages that have been found useful in areas of research. I must say, first of all, SAS. SAS is a statistical analysis system. It's a comprehensive software that deals with many problems related to statistical analysis, spreadsheet, data creation, graphics. SAS is a layered multi-vendor architecture, regardless of the differences in hardware operating system. SAS application look the same and produce the same result. The three components of SAS system are host, portable applications, and data. It's a 100 crore project and is being run at so many places in India. So this software caters almost all the needs of this analysis purpose. But it's a paid software and a very costly software. This license is being provided to the institutions, and then scientists and researchers can use it. There are three basic sections of SAS program. Data, cards, and procedure. The data section is invoked with the data statement. In the cards section, user supply the data. SAS system has a collection of ready-to-use programs called procedures. These procedures can be invoked with the proc statement. So this is something about the SAS. Notes are there, but as I am not going to show you the SAS software here, so I will also skip this, but I have to keep in mind, and everybody has to keep in mind, this is probably the number one and the first class software throughout the world. There is a SAS institution in US for this. So this is above all, from in all the softwares which are available, the SAS is the number one. There are all statistical things, I think, that has been taken care by this software. Then there are so many other softwares like JMP. It's a statistical discovery software for the Apple Macintosh, the Power Macintosh, and Microsoft Windows. JMP is a dynamic statistical graphics. It shows each statistical result graphically as well as with reports. It makes us understand the data better, along with obtaining patterns or interesting points that do not fit patterns. This can lead to important discoveries. This works on the principle of discover more, interact more, and understand more. Like SAS, JMP uses a general computational approach that works for any general linear models, balanced or unbalanced. Multivariate analysis means maneuver that has been taken care by this software. There are so many other things which have been taken care by this software. Then there is a software that's called SPSS, Statistical Package for Social Sciences. Is a very comprehensive and flexible package that consists of software tools for data entry, data management, statistical analysis, and presentation of results. The data generated from the designed experiments can be very easily analyzed for both balanced and unbalanced data. Starting with the simplest designs like CRD, completely randomized design, randomized block design, nested factors, row column designs. The general linear model univariate procedure provides analysis of variance for one dependent variable by one or more factors. We can use generalized linear univariate there for simple analysis like CRD, RBD, LSD, row column designs. Then there is a general linear model procedure. One can test null hypothesis about the effects of other variables on the means of various grouping of a single dependent variable. One can investigate interactions between factors as well as the effect of individual factors. So this statistical software, SPSS, I am going to demonstrate, and I am going to put some examples, put some problems on the in this software, and will analyze the data and will come out with the results. I will show this data. I will show this SPSS software. I will demonstrate it. And I will try to find out some basic statistics with SPSS. Then there is another software that is GenStat, has been developed by Rothamsted Experimental Station. It's an interactive general purpose statistical package. It has got a flexible command language. It covers most of the standard statistical analysis procedures and can be easily extended for newer techniques. It is particularly useful for the analysis of designed experiments. The fitting of linear models and general linear models. It also covers most multivariate techniques, time series analysis, optimization. Under the analysis of designed experiments, it covers block structure, define the blocking structure, covariate, treatment structure, ANOVA, display, ANOVA, AKIP, copies, ANOVA, REML. So this is about the GenStat. Then there is GLIM, Generalized Linear Interactive Modeling, has been developed by the Royal Statistical Society Working Party, UK. It's an interactive command-driven package which is primarily concerned with fitting generalized linear models. This means that it covers regression, ANOVA, probit and logit analysis, and log linear models. Then there is S+, a software, it's called S+, is an interactive language designed for expert statisticians. It is based on a wide range of library and functions. There is a complete tool set for mixed effect models. It's also useful for graphics. Here, one thing which is very much, which is worth noting, S+ is the paid version of our software, or we can say it's the paid version of our language with some modifications. Actually, the S+ is a software, it's a paid software. Then on the contrary, it has another software that's called R software. That's having only few modifications, some more modifications or something, some extra features are in S+, while as the rest of the things has been taken care by the R language. This R software, I am also going to demonstrate here and show how to come out with graphics and with some results through this R software. Then there is a Shazam. It's a computer program for general and specialized uses in econometrics written by Kenneth P. White. The program can be run in batch mode or interactively at a computer terminal. Shazam is designed to grow so that algorithms and procedures can easily be added by programmer familiar with the internal structure of the program. Then there is another software that's called SysStat. Here, I would like to say all these softwares, almost a trial version is available on the internet. One can go and find out this SysStat. He can download a trial version of this SysStat. He can download a trial version of Minitab. He can download a trial version of SPSS and can work with this. If he is doing a research, the trial version remains with 15 days, or sometimes there is a warning, it may go off after 15 days or after 3 weeks. But if there is a person or a researcher or a scientist, he wants to analyze his data, he can use these softwares and can come out with the results. But the R software is there. It's freely available on internet. I will give the details about the R software. So it doesn't go off after 15 days or after 21 days. But rest of the software's trial version is there. One can use them, but automatically they will go from your computer after the trial version period is over. So Systat is a software. Mostly all these softwares are having so many things in common. Systat and Minitab and SPSS almost apparently they are looking same, but there are differences definitely. Systat was designed for statistical analysis and power graphics of scientific and engineering data. It provides a powerful statistical and graphical user interface environment using descriptive menus, toolbars, and dialog boxes. Systat's initiative Windows interface and flexible command language are designed to make your research more efficient. It offers a huge data worksheet for powerful data handling. Then there is another software that's called Minitab. Minitab is a statistical package developed at the Peninsulan State University by the researchers. It's a powerful statistical software. This Minitab I'm also having with me. I will also demonstrate a little bit about the Minitab also. That provides a wide range of basic and advanced data analysis, comprehensive statistics, regression, correlation, ANOVA, multivariate, non-parametric, parametric, cross-tabulation, simulation, and it's a very user-friendly package like SPSS. So many persons, they either use SPSS or Minitab, but the condition is that these are all paid softwares, a license is required for them, they can use it. One more thing I would like to say that all these softwares, they have considerably changed from last so many years. I remember it was 1993 when I was at Indian Agricultural Statistical Research Institute, New Delhi, under the CRAB, Indian Council of Agriculture Research. We were using SPSS version 3. It was all Mino driven. Nowadays, I can say such a cumbersome and such a difficult software because we have to put F1, F2, F3, so many other things. It has changed. Then came version 7, version 11, version 13, version 17, then 19, 21, 23. These all softwares like Minitab. It has also started and there is a Minitab perhaps 26 or 27 available. There is a SPSS 27 available. That means if there is a new version, that is an improvement over the previous one. It's taking more care. So previous one, though even almost our all needs can be easily taken care by the Minitab 11 and Minitab SPSS, but still there are some researchers and some scientists, those who are working on the complex data, they need some more comprehensive and more powerful softwares. And for that purpose, these versions are being upgraded, like 27, then 29. Then I would like to say now R software. R software, R is a free software environment for statistical computing and graphics. It is similar to S language. As I have already said, there is a paid version of R that's called S+. So it is similar to S language and environment, which was developed at Bell Laboratories by John Chambers and colleagues. It is commonly used as language for data analysis and graphics. It is freely distributed under the GNU. GNU is an operating system that is free software foundation. It is distributed under the term GPL, General Public License, and can be freely downloaded via internet. And its distribution was carried out by several statisticians known as R Development Core Team. Site address via internet is http://cran.r-project.org. That means if you will just open your laptop or your desktop and you will put this site address, in your Google or on your internet, it will directly take you to the R software. There is a somewhere written on the left side of that, download. You have to click it, download it, and that software remains on your laptop forever. It's there. And if you are anytime, anywhere away from your home and you don't have a software, but you have to analyze your data, you can readily download it from the internet and use it. And again, I would say that this too has improved. There was a version 2.1, 2.7, 2.2, 2.3. It's also being upgraded, and we can now this time have a R software. Or even simple thing is that if you will just put the R language or R software in your Google, click it, it will directly take you to somewhere where you will find that download R software freely. You have to say, okay, click it. You can get R software on your laptop. I'm having this R software in my laptop. I have just downloaded it some 2-3 days before. So I will show you some basic. I would consider that as I have been told by the Dr. Joshi, that you consider that the 95% participants are very much new to the softwares, and it will be better if we will start with the very basics. I will start with the very basics in this R software, and I will just show you how this R software works and what you have to do, because this is freely available on internet. Just a minute. It will take me 1 or 2 minutes.
Sir, you are muted, sir. You kindly unmute your audio, sir.
Hello?
Yes, sir, you are
Hello?
audible.
Yes, sir. Yes,
Am I
sir.
audible?
Yes, sir.
I'm sharing my screen. Is it visible?
Yes, sir.
So this is the R software. I'm sharing this screen. It's our software. Introduction. R is a free software environment for statistical computing and graphics. R is a system for statistical analysis and graphics created by Ross Ihaka and Robert Gentleman in 1995. R is both software and a language, considered as a dialect of the S language, created by the AT&T Bell Laboratories. USA, which is the birthplace of C language and Unix operating system. One of the important features of our software is that it is an open source and freely available on website. Our language is essentially a functional language for all practical purposes of data analysis and graphics. Some important features. It's an integrated suite of software facilities for data manipulation, calculation, and graphical display. It has an effective handling and storage facility. It has a suite of operators for calculation on arrays in particular metrics and data frames. It has a large number of functions for data analysis. It contains numerous graphical facilities to display. It has its own programming language. So first of all, what you have to do, go to the web page, the left part, you will find CRAN and click there. So left part of this, it's here, CRAN. Here is written download. If you will just click here, you will have our software. It will take 2 or 3 minutes or 4 minutes, and there will be an icon also on your desktop as our software and its latest version. Along with it, that's the latest version, our software available on the internet. Just write down this thing on your Google. CRAN is here, download it, and you will be having our software on your laptop or on your desktop. This you will be getting first of all. Once you will double click the icon or software on your screen, you will get this. R version 3.2.2. This is the latest version this time. R is a free software and comes with absolutely no warranty. You are welcome to redistribute it under certain conditions. R is a collaborative project with many contributors. Now, let me come here. This greater than sign is called R prompt. Once you will double click the R software on your screen, you will come to this greater than sign. This is called, and that too in red color. This is called R prompt. Now, before executing certain commands on R software, I will tell you this is called R prompt or R command, this greater than. This is called assignment factor. This is less than dash. C, this is the function. C, then small bracket, big bracket. Then this is used for vectors. Hash is nothing but explain statement. Say, for example, you are putting F and T, and you know that F stands for false and T stands for true. You can write down hash, then you will write down the statement F stands for false and T stands for true. C is called the concatenation. Now, EDA is the exploratory data analysis. This is the OR console or OR environment. You will be having this here greater than sign. It comes automatically. And if you will write down 6 plus 8, enter it, you will get 14. And if you will write down 6 star 3, that means multiplication, you will get 18. If you will write down, again enter. 6 minus 8, you will get -2. If you will write down 15/3, you will get 5. If you are having 100 --45, it will just take the observations from 100, 99, 98, 97 up to 45. Now let us come here. We are now assuming some values. Let us write down X. As I have said that this is the OR prompt. This is the assignment factor. X less than -76. That means X takes the value 76. Enter. Y less than dash. It's 63. Out. Assignment factor again less than dash X plus Y. Enter. Out. You will get 130. That means it has taken X plus Y. That is 130. Or you can say out X star Y. That means it has multiplied 67 by 63. And the answer is 4221. Out X minus Y. It has taken the subtraction of these and the answer is 4. X divided by Y. The answer is this. If then you are having X and you are giving assignment factor, that means assigning value to the X like 2, 5, 6, 7. And then you will write down the mean X. You will get the mean. If we will put var X, you will get the variance. If you will put the SDX, you will get the standard deviation. And if you will put the summary X there, you will get the minimum value, maximum value, first quartile, third quartile, mean and median. Now let's take instead of this only 4 observations, we will put down X and say 15 or 100 or sometimes huge amount of observations. We have to just write down them and put the mean X that will give us the mean, or write down the summary X that will give us the summary of the data. I will show all these by doing here in our software. I'm showing this slide, but I will show it practically also. But before that, I would like to have another slide. We are having weight, assign its value. Let this be the weight of something, some items. 89, 94, these are the weights. Mean weight, it will give us the mean weight. Then standard deviation of the weight and variance. Then we can have median weight, coefficient of variation, quartile, interquartile range, minimum, maximum, range, difference, whatever we want, we can write down here with this red color. It automatically comes in red color and you will come out with the result. Then we are having the principal graphic techniques used in R. Bar chart, bar plot, histogram, pie chart. I will show you now our environment here. I will share that screen again. Our software should give it R. This is R. Where is R? Yeah. I'll show it on the screen. Minimize it. It's there. You minimize R. I'll show you. Minimize colors. This is here. This one. Yes. I have double click it. Is the screen visible?
Yes, sir.
Is it visible there?
Yes, sir.
So I have just double clicked that icon and this is my screen now. R version 4.101. This is the latest one. I have just taken it some 1 hour before. I have downloaded it. R is a free software and comes with absolute no warranty. This is now, let me start from here. What I have said already there in my previous slides. Just a minute. Please. 6 plus 8. Is it visible? Hello?
Yes, sir.
I will write down here 6 plus 8.
Yes, sir.
I will enter. It's 14. I'll write down 6 as I have showed in my slide, star 8. It's there. I'll write down it's 56 hash 9. I'm dividing 56 by 9. Enter. Result is 6.2222. So this is the simple what we can do with the help of calculator. Now this can be done here. Now I'll write down. थोड़ा सा आगे आता हूं मैं इससे. Again, X, I'm saying less than slash C, and I'm putting certain values for X. Say 12, 13, 23, any value, 14, 27, 24, 56, 78. Close it. Enter. I can write down the summary. Say, I will write down SUMMARY, summary. Summary of X. Enter. This has given me the summary of the above values. The minimum value is 12. Maximum value is 78. First quartile is 13.75. Third quartile is 34.25. Median is there. Now, a little bit knowledge about the statistics. There are some measures of central tendency. Those measures of central tendency actually means those measures which tell us what is happening at the central part of the values. So these are certain measures, they give us an idea about the central part, like mean, that tells us that the mean of the above observations is 30.88. Median is a positional mean, means it's a positional average. If you are just arranging the observations in ascending or in descending order, you are getting the median as 23.50. But why this is 23.50? Actually, there are two medians. These are 1, 2, 3, 4, 5, 6, 7, 8. 8 observations. That means middle observations are 4 plus 5. That means 23 plus 24. Then it has taken the mean of these two. It is 23.50. First quartile means that this gives us a, I don't know what's happening. Somebody is just doing some mischief. Please. Hello? Hello? Is it
Yes, sir.
audible? Somebody is doing some mischief. I think he's putting some hash or something.
Mike, sir, please remove. Yeah.
Okay, let him do his job, but we will do ours. So I think that first quartile means that it is the only 25% of the observations. 70% or 75% are on the other side. Third quartile means 3/4. That means 75% observations. Left are 25% observations. This gives us an idea about the central value of the data. Mean, median, first quartile, third quartile, they are called measures of central tendency. They give us an idea about the central part of the distribution. Now, let me again proceed further. I will write down here weight, W-E-I-G-H-T, weight. C, then certain values. I have written certain values there. I will put this. I will say the mean, M-E-A-N, mean. What mean? Mean weight. By entering, I have got the mean weight is, out of these observations, the mean weight is this. I can again get the summary, S-U-M-M-A-R-Y, summary, weight. This is the summary statistics. Now I will show you something like that, BAR, bar plot, P-L-O-T, bar plot weight. I have got the bar plot. Is it available on the screen? Hello?
Yes, sir. Yes, sir.
We are getting the bar plot on the right side of the screen. That means it's also having a good graphic facilities there. This is the bar diagram of the values which I have put for weight. I can have the histogram, H-I-S-T, sorry. I will write down hist, H-I-S-T, hist, W-E-I-G-H-T, weight. Enter. This gives the histogram. I will write down the pie chart. Just a minute. Sorry, pi. Only I have to write down the pi. This is the pie chart, which you might have seen on at so many places. These are the graphical representation of the data. Bar chart, histogram, pie chart. I will write down the box plot. Box plot, W-E-I-G-H-T, box plot weight. This is the box plot. And if some of the researchers will remind that many of the research papers are having a bar diagram with it, then this box plot. This box plot again conveys, this is a complete description about the summary statistics. This box plot tells us this is the minimum observation, and you will see that the minimum observation above is 12. So this corresponds to 12. Then this is the maximum observation. See from the data, the maximum observation which I have roughly put here is 78. This gives the minimum observation. This is the maximum observation. This is the first quartile. As here mentioned, first quartile is 36.75. This means first quartile means that only considering 25% observations, left over are 75. This is the median. Median means the positional average. This puts the 50% observations above it and 50% observations below it. This is the, remember, median is the positional average. Sometimes we don't need quantitative average. We don't need a mean, but we need median. Median is that value of a distribution which divides it into two equal parts such that the observations above it are equal to the observations below it. So this is the third quartile, this much. You can see the value there in third quartile, it is 55.25. This is the median. So this box plot, why there are on research papers number of times this box plot? Because this box plot is a good description of the graphical display of the data. A statistician or a scientist or a researcher, by seeing to the box plot, can easily identify what was his minimum observation, maximum observation, middle observation, first quartile, third quartile, and median. Though it doesn't give us that much description here, but as a statistician, I am telling you these things. Again, we are having QQ norm. QQ norm of weight. This gives us the scatter diagram of the above values. You can see that 12, it's here. You can see that another value, some 30, I must say what's 34. There are two observations of 34. Fortunately, I put some two observations of 34. You can see that 34. This is the scatter diagram, dotted diagram of our data. This is being driven by QQ norm. We can say QQ plot. If you are putting above weight, it's weight. If we will take X into consideration, it will go about the X. Let us put weight, W-E-I-G-H. Sorry, this has not taken this statement. Something has went wrong. When something is going wrong, it says us error in sort. Argument Y is missing. Something is wrong. I will just find out that what was wrong. It is QQ line I have to write. QQ line, not QQ plot, QQ line. Now this is the best line which we can draw through the data. Now let me again share the screen. I have shown this, the principal graphic features used in R. Is it visible there? Hello?
Visible.
Now we can have another thing. We can have t-test, chi-square test, z-test, correlation regression, the basic statistical tools. We want to find out the t-test. We want to find out the t-test for two sample means. We want to find out the correlation. We want to find out the regression. I will work out that with the help of this software here. Then I would like to show you one thing more. Length, L-A-N-G, what is the spelling of length? L-A-N-G-T-H, length. Length weight. That means length weight means what was the length of weight? How many observations are in weight? You can say that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That means perhaps we may not have missed some observations if we'll put the length. That will give us the whether we have observed our, whether we have inserted our data, put our data rightly, or there are some observations missing. We can find out that by length. Now let me say that I will show you X, C concatenation. I will put down some 7 or 8, 12, 13, 30, sorry, I must say 15, 16, again 15, 16, again 17, again 18, then again 21, then 22. These are the values of X. Enter. 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7, 8, 9 observations for X. Then I will put 9 observations for Y. Six, 7. I have put 9 observations for X. 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Say X was something and Y was something. Say X was the height of the person and Y is the weight of the person, or X is anything, any variable, Y is another variable. I want to find out the correlation between these two variables. What is correlation? Correlation means numerical relationship between two variables. We can say that if one variable increases, another increases, that's a positive correlation. One variable increases, another variable decreases, that's a negative correlation. And if one variable increases or decreases and another variable increases and decreases correspondingly and accordingly, that's the perfect correlation. Correlation means change of one variable produces change in the another variable, whether these two variables are correlated or not. I want to find out that X was a variable, Y was a variable, and find out the correlation. In so many research papers, we are being asked to find out the correlation between this height of a person and then its weight. Find out the correlation between head circumference and chest circumference, whether it's chest circumference which increases and then head circumference increases, or they are independent of one another. So what I have to write down, I have written X, I have written Y, I will say enter, then core X, comma Y. This is the command. I will enter. I have got the correlation. There is a correlation of 0.369. That means X and Y are positively correlated. And we know that the coefficient of correlation, it ranges from -1 to +1. If we will put the same data, but if we will put the different data where one variable increases and another variable decreases, we will see that the correlation will come negative. I will show you that example also. I am using you know that on the right side of our desktop or laptop, we are having 4 arrow keys, right side, left side, up and down, 4 scroll keys. We can use that also. I'm using that. I'm putting it up, I'm getting 4, I'm again putting it up, I'm getting the second variable, I'm again putting it up, I'm getting the X. Now I have not written X, I have copied, say, X from there, and I have entered. I will write down the Y. But I will write down the Y in such a manner actually to tell you that whether it finds the correlation in the perfect. When it is 12, say the value of Y is 9. When it is 13, the value of Y is 6. When it is 15, the value of Y is, I must say, 4. When it is 16, the value of Y is 3, 3. 16 and 16. When it is 17, the value of Y is again 5. When it is 18, the value of Y is 3. When it is 21, the value of Y is 2. When it is 22, the value of Y is 2. There are 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9 observations. 1, 2, 3, 4, 5, 6, 7, 8, 9 observations. We are having observations for Y. We are having observations for X. I will enter. Then I will write down again core X, Y. I will just enter it. I have got a negative correlation. That means X variable and Y variable, they are negatively correlated. We can find out t-test, we can find out z-test, we can find out chi-square test, we can find out regression. I will show you how to find out the regression. I will write down for regression, the command is, say we are having the same values for X and same values for Y. I am considering this X and this Y. Now I will show you the regression. The command is fit. We are generally saying that fit a regression line or fit a regression curve. We will say fit, then assignment, then LM. LM stands for linear models. Then I will say bracket start X. And if you will see on your desktop or laptop, if you will go to the extreme left, where you will find the escape key. Down the escape key, there is this notation. You might have number of times seen this notation, it's there. I have put it this. That means X corresponding to Y. I will just close it. This is, I am fitting a regression line between X and Y. I will enter. I will say fit. Sorry, I will say summary fit. I want actually the complete summary of this regression. Summary, FIT, fit. Enter. I have got the complete description about the regression. Minimum, first quartile, third quartile, median, maximum, coefficients. In regression, we are having Y is equal to beta naught plus beta 1. Y depends on X. This is the intercept, means beta naught. Its standard error, its t value. This is the y. It's beta 1, its standard error t value. And this is the significance. This is the multiple R square, adjusted R square. R square tells us how far the dependent variable is being explained by the independent variable. This is a complete p value. And definitely all of you might be knowing that any p value which is less than 0.05 is taken to be statistically significant. It's a probability value which says that any p value, if it is 0.04, it means significant. If it is 0.008, it means again significant. 001, it is again significant. 2, it is again significant. But if it is 231, it's not significant. Any p value which is less than 0.05 is taken to be statistically significant. Likewise, we can find t-test. I will just skip all those things because I would like to show you also some this SPSS, because SPSS is also very user friendly. But I don't think that I can show you many tab then. But I would like to show you this. But I will tell you there is a command for t-test, t.test. If you will write down t.test x, you will find out one sample t-test. If you will write down t.test x comma y, this is comparing the two. We are comparing population x, sorry, sample x with sample y. We are comparing this. If you will enter, this is the two sample t-test. Two sample t-test. It's here, two sample t-test. This is the t value, degrees of freedom, p value. This exponential minus 0.07 means highly significant. So we can find out chi-square, we can find out other things. We can go for ANOVA, but I will just again go to my slides because I would like to show you. This is just the practical demonstration, but I would like to show you that this is my, again, I don't know whether new share, may I have to show you what I have to show you. I have to show you this. Is my this screen now available? Some basic test statistics by R. Hello?
Yes, sir.
Okay. Because I am moving from one slide to another, one place to another, I have to. This is some basic test statistics which I have shown: length t-test, two sample t-test, correlation, regression. Then there are analysis of experiments which can be carried out by R language and R statistics. Little bit थोड़े difficult है, but उतना भी नहीं. But it cannot be a whole or cannot be understood in one day. But if I would like to say one more thing, what I have done personally, this is my personal experience. I would like to see my face. Would you like to allow me first of all show my face? I want to show you something.
You have to.
I want to see my camera on my side. Hello. Share the screen for camera. I have to new slide. Where is the camera screen? I mean the basic screen. I don't know how to get that now. Oh,
From here. Just do it.
Share. How it is possible that it's there? I'm there. What I have done one thing. I will tell you. See, I have already learned some 3-4 days R training, and I have learned something from R. Then I have taken out a complete printout from the help menu of this R software. That printout is with me. I'm showing you, this is the printout. I have written by my hand here R. This is something. I have taken out the printout. This is a complete manual for all available in R software. I have taken out this manual. These are roundabout some, I must say, 90 pages. And if one will have this printout with him, he can take out the printout from this R software, a manual, complete manual, as we are having a manual for our LED, LCD, and other things. We can have a manual. Invoke the R environment on your screen, take out the manual, start from very basics, and definitely after 2-3 days, or 2 days or after 3 days, you will be reaching to designing the experiments, and you will be reaching to probability logic and another thing. But this is, I think, that the simplest procedure. Take out a printout of this manual from the R software, have it with you, and try to resolve the things. In case you are having some difficulty, as this nowadays all these softwares are user friendly, help manual is there, and presentations are on Google also there. You can seek the help from all these things. And I think that within 6-7 days, you will be having complete, you will be very much well versed with this R software. Depends on one's intellect. And sometimes it may take months, but if somebody is very much interested, and if he will put a lot of amount of his brainy work for this, he can do it within a week. This is my software. Simply, I have taken out the printout of this manual, of the manual fonts. Now let me start again, share my screen regarding the other presentations. Is it again visible, my screen? Analysis of experiments.
Yes, sir.
So this is the analysis of experiments. This is the very basic and the first experiment, that's called completely randomized design. When we have to we are having treatments. What do we mean by, first of all, treatments? Treatments doesn't strictly restrict us to treatments. Treatments means anything which is under comparison. Sometimes there may be different procedures under comparison. For an engineering experiment, there may be different automobile machines under under comparison. For medical point of view, there may be different drugs and different treatment lines under comparison. For agriculture point of view, there may be different fertilizers, manures, or different fertilizer doses, depending on the subject from which the problem is. Treatment means whatever is under comparison, they are treated as, they are called as treatments. If we are simply having some treatments, we have to make a comparison of those treatments. We use CRD, and if they are under homogeneous conditions, they are in a homogeneous environment. So this is just a model. I will start from here now. We are having here 7 treatments. This is something, an agriculture experiment. We are having grain yield of rice resulting from use of different foliar and granular insecticides for the control of brown plant hooper and stem borer. These are the diseases, brown plant hooper and stem borers. We are using 7 different treatments and see whether out of these 7 treatments, they are at par, or some treatment is much better, or 2 treatments are better, or some treatment is better followed by another treatment. To scrutinize all those 7 treatments, we are performing an experiment. And you know that in any sort of statistical experiment is not taken on one observation. It has to be taken on replications. We are having treatment number 1, and we are having 1, 2, 3, 4 applications. We are having treatment number 2, we are having 4 applications. But here in this very exercise, we have kept the number of replications 4, and our treatments are 7. This experiment can be any sort of experiment. There can be 7 different drugs for inducing sleep in the patients. That can be a medical experiment. There can be then replication. जब उसकी पहली replication हुई, this was the result. दूसरी replication means repetition of the treatment. So तीसरी replication, चौथी replication, then we can say you that we are having this R environment, R console with us. This is there. What we have done, we have put the replication first of all, 1, 2, 3, 4, 5. How many replications were there? There were 4 replications. How many treatments? 7 in earlier slide. So this is the treatment 1, treatment 2, treatment 3, treatment 4, treatment 5, treatment 6, treatment 7. We are having 7 treatments. This is their replication number 1, and I have fed their data here. Then I have taken again treatment 1, treatment 2, treatment 3, treatment 4, treatment 5, treatment 6, treatment 7. This is the replication second. This is the data. Then again, treatments, again replication. Finally, we are having 7 treatments, 4 with 4 replications. That means the overall data is 28. Since the number 1 observation is replication, treatment, and yield, I have written replication, treatment, and yield. That's why the serial number here is 29. Otherwise, these are only 28 observations. 28 data points I have taken from this very slide. I'm here now. What I have to do, I will move to the another slide. These, whatever is written in red color, they are the commands, and this is crd dot read dot table, likewise this, and we have finally come out with the 7 treatments. Unka mean kya hai, unka standard deviation kya hai, lower limit kya hai, what's the upper limit, what's the minimum value, what's the maximum, and what is the least significant difference. Least significant, I'll write down it. Least significant difference. 452.7. That means those treatments where the difference is more than 452, they are different. If the difference between the 2 treatments is less than 452, they are statistically non-significant. You might have heard the term statistically significant, statistically non-significant. Statistically significant means when the difference exceeds the critical difference, or LSD, least significant difference, they are statistically significant. If the difference between the 2 treatments doesn't exceed this, they are non-significant. Then there are experiments where I will show you RBD, where we are having 2 sources of variation, treatments and blocks. Since the material is not homogeneous, we are first blocking it. That means putting the material in homogeneous groups. That means there are 2 sources of variation, blocks and treatments. Those who have read statistics or some basic concepts of statistics, they know randomized completely blocked is how to go for the analysis when there are 2 sources of variation. This is the data. These are the commands. Then this is LSD, least significant difference design. These are the commands. This is again, these are the commands. Factorial experiments we can do with this R software. These are the examples I have taken from the agricultural side. But whether the example is from agriculture side, medical side, engineering side, or any field, actually we are concerned with the data. Our main, this outcome is the data, and we are dealing with the data. Data may be anything. It may be from any field of from any discipline. This is something about R software, but now I would like to share the screen for SPSS. I hope that I'm running short of time. Stop share. Where is SPSS? It's here. Share where from? I have to share it. It's here. I have to share it. I don't know what I'm doing. What's wrong? Hello?
You can share the thing.
Yeah. Now it's okay. I think so. I have to. Kuch to hai. This is my share again on? Am I sharing my screen?
No, sir.
So I think that there is something which I have to do. I will see that. I have actually closed that. I think so. I have to again invoke my this. Just a minute. Give me 1 minute. I will see. Oh dear, I have to screen this screen, share now this screen. Hmm. Hmm. Share screen is there. I have to share this one. Yes, yes. Share. Is it visible now?
Yes, sir.
So this is statistical software. I have shown you, or now I will show you something about SPSS. I will skip all these things. I will directly come to the features of SPSS. I will skip. It's easy to learn and use. It includes a very user-friendly package, SPSS, statistical package for social sciences. And it's finding it's very much importance in biotechnology, medical, and so many other fields. SPSS, a very user-friendly name in so many institutions. But again, paid software. SPSS editor, data editor, syntax editor, pivotal table editor. This I will, I don't know, I'm not getting that much of that. I will show you the final. This is something. This is what once we are having SPSS trial version, which is available on our internet, just download SPSS trial version. We will find out an icon on our desktop, double click the icon and we will come to this. This is called the SPSS data editor. Then again, like as we are having in Word or in Excel, file, edit, view, data, transform. I will show you all that. Okay. This is the SPSS data editor, just like we are having in Excel rows and columns. We are having two things here, data view and variable. This time, this is the data view. We can just put any numerical data, 11, 10, 13, 14, row wise or in column wise. Is it okay? Then if you will click the variable view, see what happens. Sorry. Okay, sorry. If we are just clicking on the variable view, this will come. Now it will name, type, width, decimal label. That means we can name that column now. I can name that by weight. I can name another column by length. I can name another column by circumference. I can name another column by, if I am from the medical field, systolic blood pressure, diastolic blood pressure, heart rate, SpO2, so many other things. Depending on the field where from the observations are, I can name them. Then there can be the type of the observations, whether they are numerics or they are they are not numerics. They are like gender we are having, male and female, but we can code them as 0 and 1, 0 for male and 1 for female. What's the width of that decimal wise? Decimal means if you are having 22.34, you can write down it. But sometimes there is a very sensitive data which we have to take care of up to 10 decimals. That means 22.10456823. After decimal, there are 10 variables we can change it. We can label it, value it, missing values, column. I will show you that. What's the next? Dialog box. Most sections open dialog box. We are having now, I have put in first column PP, pH. This is the PP value, pH, NH, yield. And again, I will go to the income. This is income. I can have income in numeric value. Income can be numeric, thousands, lakhs income, but gender, it is not numeric. It's male and female, but I can put it here too. That means I will show in the next column that what I have done. I have put, sorry, that I have skipped. I have put instead of male, I can put 1, and instead of female, I can put 2. I can decode it later on. Or for colors, we can have blackish, yellow, blackish, or I must say white colors. I can put 1, 2, 3, 4, decode them, code them, and later on we can decode them. Now file, you can have all those which are having in our Excel. New open database like this. I have put the data in columns now for those 4 variables. If you are having 4 variables from any discipline, from any field, you can put the data. Simple, the way you are putting the data in Excel. Then you can open the file. This is the data, you have opened the file. Then edit. This is the second one. This is the file. This is the second one. We are having data. I don't know what is happening. Actually, I'm getting. This is the second one. This is the numeric, which I have earlier skipped. I was telling you that. This is something. Income, gender. Here for gender, I have put 1 and 2. 1 for male and 2 for female. It's there here. Then data. We are having so many subfiles there. Then for transform. Analyze. Analyze me, we can go for descriptive statistics, mean, generalized linear models, correlation, regression, log, linear, classify. No command, nothing you have to write. Just open that submenu and that will lead you to the another menu. Open the window, fill out the requisite column and just get the results. Gallery, utility, windows. This is topics. In help, we are having topics. If you are getting something on the screen and you don't know, you go to help, open the topic, that will give you a way in a very easy language. Mein aapko bata dega that what does the topic stand for, topic mein kya hai, how you will go and study that topic. This is the tutorial where you can teach yourself what is chi-square test, what's t-test, what's z-test, what is correlation, regression, so many other things. Self-explanatory. Now, basic steps in analysis. Get your data into the data editor, select a procedure from the menu, select variables for the analysis, examine the results. We are putting a variable like this from 1 to 16. Height ke humne variables dal diye from 1 to 16. We are opening add-ons here, bar, 3D bar, box plots. So this is the graphical features. This is the bar. I have put that here. Utility, I have gone to the bar. I'm simple bar and clustered bar. I am putting that height from here. It was height. I have just clicked this. This has put the height into this very box. And now I will say again, okay. Or I will put it here. I don't know what is happening actually. I'm in a hurry. That's the reason. This is the box plot. Sorry. This is something. Height. This is the box plot of the height. I have selected their box plot. Now I will select a histogram here. And that will give you the histogram. Now this is the some variables are with me 30, 32, 35, 38. And I want to find out the summary statistics. These are the variables from any discipline. We can just go to the analyze descriptive statistics, then descriptive. Open analyze, click analyze, find out descriptive statistics, then go to the submenu descriptive, select it, yield. We will find out the mean, standard deviation, maximum, minimum, other things. This is the descriptive. Minimum, maximum, mean, standard deviation, variation, skewness, curvature, measures of central tendency. These are the variables. Seed yield per plant, plant height. Seed yield per plant, plant height. Seed yield per plant, plant height. Find out the correlation. What I have to do? put the data in X and put the data in Y. Just go to the analyze, select correlates, bivariate correlation, enter it. This will be finding here, and after clicking this, they will just move to this very place, seed yield and plant yield. Is there any correlation between the seed yield and plant yield? I will just click OK. This gives me the correlation. It's here. 0.287. That means positive correlation. Again, I can find out the regression. Just go to the analyze. Open the menu, you will go to the regression, go to the linear regression. You can have binary logistic regression, multiple, this multinomial regression, probit regression, probit analysis, log linear. Just see that. You have to select one dependent variable, another independent variable. This is the outcome of this. And sometimes if you are not understanding the outcome, just right click it on that visible window and you will find out the explanation for that. What does this output stand for and what does it convey? Sorry, I'm not getting it sometimes. I don't know. You're doing things in a hurry. This is multiple regression, multiple regression. This is one sample t-test. This is t-test. This is two sample t-test. This is output one. This is two sample t-test. Do you know what's difference between t-test, two sample t-test and peer t-test? We are having the most commonly these terms are misused. There is a two sample t-test and peer t-test. Two sample t-test means we are having two independent samples, we are comparing them. We are having a sample from one place and sample from another place, we are comparing them. But peer t-test means that we are having observations, we are having individuals, we are having subjects prior to something and after something. That means if we are having some 30 patients and we are recording their systolic and diastolic blood pressure and give them an injection and record their again systolic and diastolic blood pressure after 2 hours or after 3 hours. Subjects remain the same, but readings are taken pre and at post. That means once the subjects remain same, they are called paired t-test. Say, for example, there are some persons, they are being given a training. Let us take this. There are so many participants which are attending this training, but we can have a peer t-test. Now, how? We have asked them certain things, certain questions about this SPSS and R. Perhaps some of them were able and some of them were not able to answer. But after getting this 1.5 hour or 1 hour training, we will again put them on some more questions. Whether this training has really imparted some significant impact on the trainees, we can perform a peer t-test because the subjects remain same. But if we are taking the subjects from this very place and the same thing is going on at another place in another university, we are comparing those, then it is a independent two sample t-test. But if the subjects are same, very much which are attending this session this very time, we are having them some questionnaire pre and post this training, they are called paired, they will fall under paired sample t-test. That's why I'm giving a little bit explanation about the things. We are having t-test, we are having, this is chi-square association. This is then design is CRD, RBD. I think that we have already crossed some 10 minutes. Two factor experiments. I am also having Minitab with me, but I don't think that's similar to SPSS. Now, I think that I must close it. What's the opinion of the audience or how far it's been taken? Hello?
Yes, sir.
Is it over? Should I conclude it? Or
Yes,
should I
sir.
go for Minitab now? Otherwise, I think that there may be so many participants for them. It might have been boring for them because I'm telling them something about, but this is just a sort of pudding to the beginners and to those who are not touching the softwares. If they are having any problem related to their discipline, it may be engineering, medical, it may be agriculture, it may be veterinary, it may be social sciences. They are having their data And I must say that there is a well-known saying. Today, scenario is that we are floating in the ocean of data, but we are striving for the information. And here comes the role of softwares. Softwares are used to extract the information from the data. Otherwise, we are having data and we are not expecting information from it through statistical rules and techniques. It's of no use. So to convert the data into information, here comes the role of statistics as well as the role of please.
Thank you, sir, for giving the most valuable insights on the topic use of statistical software in research. We hope that this information will help researchers during the software selection process and motivate them to provide complete information about specific software employed in studies. Now we will go ahead and take up a few questions now from the participants. The first question is, are software similar to S language? What is S language?
S is actually a software, or S language is there. I don't know much about that, but I know only this thing, that there is a similarity between R and S. I have used S software, S language. Then it was told to us, it was earlier in a paid manner, and we were then told that stop it in your institution because we have to pay some amount for this. This resembles to R software, and you can use R software, but no doubt if something is paid, it's very well understood that definitely it will be having some advantages over this R software. Not beyond this, I don't have any much knowledge about this.
Sir, I have a question.
Please.
Sir, can I use R software? Can I use R software to plot double x-axis and double y-axis graphs where I am having more number of values? How I can use it in a short time?
Yes, why not? You can use our software and you can write down the commands yourself. You can write down even the programs yourself. But for that purpose, it needs that a person must be, I must say that well versed, or he must be knowing how to use this R software. Actually, the aim of my lecture was to give you the very basics, but no doubt this is not the limitation. So R software is a very comprehensive and a very nice software. We can even, it is having a facility that we can write down the programs in it. But the question is that whether a person knows how to proceed further. And it will take definitely, if a person is well versed with our software, you can have, I think that most of the things done through our software. But I would like to say one thing, important information for the participants, that our software is having a disadvantage. As per my opinion is concerned, it is a case sensitive software. If you are sometimes writing capital X and in next step you are writing small x, it will show you an error. That means you have to take care of that. It's a case sensitive software. That's a lacuna with this. मतलब आज की डेट में केस सेंसिटिव होना चीजों को it's a little bit worrying. So sometimes I'm using capital X and capital Y, and in the next step I forget to use the capital X and capital Y. I'm getting the error. But once I'm getting used to this software number of times, I know all these things. Please.
Sir, modeling. I can also do modeling with the help of R software.
Yes, of course. I have secured so many things. Modeling is there, not only modeling. There are linear models, there are generalized linear models, there are non-linear models. We can take care of all these things through our software. Actually, I must say you one thing. I don't know how people will take it and how the audience will take it. If we will just take our Excel, which is the very basics with every computer, I must say as a person, I accept this. As a teacher, I accept this. It is totally unexplored. Only 10% or 5% or 20% is explored. Excel itself gives us so many things. We can do CRD, RBD, correlation, and so many other things in our simple Excel, but it is unexplored. People are not using it, or they don't have to use it, or they think why to go to Excel because it will be a little bit cumbersome. Why not to go to our software directly? It's likewise in our software. There you can do so many things. I think that perhaps जितनी हम लोगों की need है शायद हो सकेगा. But I will tell you where these softwares actually are used. These are used in very complex situations when we are having metadata. I was earlier telling in the very beginning, metadata is the term where we are having data about data. We are having a single plant or a single individual. And say, for example, we are having 10,000 individuals now, and a particular individual is having some 70 data points. He is having his TLC, DLC, heart rate, systolic blood pressure, diastolic blood pressure, SpO2, his urea, creatinine, and so many, all these variables. They are associated with a single variable, and we are having 10,000 variables. Now you can see there is a chunk of data. Handling such situations, definitely they cannot be done by the calculator, or they cannot be done by the manually. No doubt, if there is an intelligent person, he can do that. I'm not denying that, but it will definitely take him months to resolve that issue. But they can be easily taken. You have to just feed the data and follow certain procedure and follow certain things and follow certain commands, and you will come to the conclusion with the results. I will tell you one thing. I am going to show you one more slide at the end. I hope that Madam will allow me. I am going. I have
yes,
to put that.
yes,
I'm showing you just one slide. It's a slide, nothing more than. Just Just a minute. I will call the help person. This is the slide which I have shared. This one. I have to show them to this. I have shown them this slide. I want to show them. Is it visible? I have shared some slide. Is it visible?
No, sir.
It's
No, sir, not visible.
Not visible.
Okay, it will take me a while. Just a minute. It will take me. Let's go down. See. Okay. This one. Is it now visible?
Yes, sir.
So this is something what our university has done in 2013. Noori, the world's first cloned Pashmina goat, produced at Sher Kashmir University of Agriculture, Science and Technology using simplified technique, is being showcased along with the laboratory of Department of Animal Biotechnology. It's here at the Republic Day 2013 of Jammu and Kashmir at Rajpath. This is Noori on Rajpath. This is, I am having that slide of Noori also with me, but it will again take some time to show the slide of Noori. Noori is the first Pashmina goat, that too cloned Pashmina goat. Why I am showing you this slide? This work has been done by the biotechnology people. They have genetically engineered this Pashmina goat, which is only found in Ladakh region in a very cold temperature. They have prepared this Pashmina goat in Kashmir in the Department of Biotechnology. So for such purposes, when you are doing some sophisticated work and sophisticated things and working on so many things, you definitely need and seek the help of softwares. When you are having something significant to show to somebody. This was shown in 2013 at the Republic Day 2013 in, this is at Rajpath. Thank you. I'm having the slide of Noori, also the world's first Pashmina goat, but it will be again time consuming. Anything more?
Sir, as we are short of time, so we are not able to take all the questions. We We will convey all the questions of the participants to your email
I have
ID.
one thing I have in my slide, my email number is there, my phone number is there. If I can be of any help, I can be contacted at tariqraja@fukaskashmir.ac.in or rediffmail, tariqraja@rediffmail.com. My email address is there. Just wait.
Now we will end the,
Just a 1 minute, 1 minute.
yes, yes.
Perhaps somebody may need because we are here to help. These are my email addresses. Not this. I will show them. This, only this. This is my email address. Perhaps I can be of some help. Is it there on the screen?
Yes, sir.
So if I can be of any help, it will be my pleasure. Because knowledge is only to share something, and I don't know much about the things, but whatever I know, it will be my pleasure to share with others.
Now we will end the questions session here. Everyone, please switch on your video so we can take a screenshot.
Okay, sir. Switch on your videos. जरा बेटा इसको वीडियो बंद कर दो मेरा. नहीं, I don't have to open my video.
मैंने आपको अभी को बता उनको बता
I'm seeing on the left side of this, there are so many things. Excellent. So much knowledge in this.
It's time for group of thanks. We have just now enjoyed a very good presentation addressed by Dr. Tariq Rajasan, an eminent scholar. On behalf of the organizing committee of the FDP, I, Amita Joshi, extend a very hearty vote of thanks to our eminent speaker for his valuable presentation. Dr. Tariq Raja is a multi-talented personality. His presentation was very informative and useful for our research work. I am again thankful to him for sparing his valuable time from his busy schedule and sharing his deep knowledge about the use of statistical software in research with us through this presentation. We all are enlightened by his valuable knowledge and information. I am thankful to the participants who joined from all over the country and made this event a great success. I am very much thankful to Architect Achal K. Chaudhary sir, President IPS Academy and Chief Patron of this FDP, Dr. Premlata Gupta ma'am, Principal IPS Academy and Patron of this FDP, and Dr. Pradeep K. Joshi sir, Head of Mathematics Department and convener of this FDP to guide us and also inspired us to do the best. I am also thankful to all the departmental heads and my colleagues and staff who work behind the scenes to execute this FDP. Our technical arrangement team, once again, thank you all for making this program successful. Thank you once again for joining us. We all will join back sharp by 2 p.m. for the next session with speaker Dr. K.K. Verma, retired Professor Rani Durgavati University, Jabalpur. Thank you everyone for your time. Goodbye. Thank you, sir. Thank you once again.
Thank you very much. Okay.
Thank you, sir.
My pleasure. Where to leave this now?